Hello everyone and welcome to my lecture video. In this video lecture, we will discuss different commercial sources of enzymes. I will cover following topics. These are enzymes from animal and plant sources, enzymes from mammalian cell culture, and enzymes from microbial sources. First of all, what is an enzyme? Without enzymes, there would be no life. Enzymes are the worker bees that make things happen. Enzymes act as biological catalysts. They accelerate biochemical reactions within our cells and affect every function of the body, from digestion to breathing. Functionally, enzymes facilitate cellular reactions that may not otherwise occur. By lowering the threshold of energy required for those reactions to take place. Some enzymes are even capable of reversing a standard reactionary response by sufficiently lowering the required activation energy enough that the reaction favors the opposite direction. Tens of thousands of different kinds of enzymes are believed to exist in the human body, each with a specific purpose. There are three general categories of enzymes: digestive enzymes, metabolic enzymes, and food or plant enzymes. The digestive enzymes category consists of the enzymes produced within your own body to help break down food into its basic components for digestion. Metabolic enzymes are found throughout our entire body, in our organs, bones, bloods, and even within the cells that produce them. They function in support of our heart, lungs, kidneys, and brain. Food and plant enzymes are naturally present in raw food. They generally serve the same function as digestive enzymes, but these are the enzymes that we may take in through our diets, as opposed to the ones our bodies produce. We can obtain these enzymes through eating fresh, raw, and uncooked foods like fruits, vegetables, eggs, unpasteurized dairy, meat, and fish. The modern diet generally revolves around processed and cooked food. But these processes destroy the naturally occurring enzymes contained in the food. This places a heavy burden on our bodies to subsidize the enzyme requirement for breaking down that food. Raw food contains the necessary proportion and types of enzymes required to digest itself. This remains one of the biggest benefits of a diet centered around raw food. The major components of the food, that is sugar, protein, starch and fat, and their respective caloric amounts determine what type and quantity of enzymes are also present. For example, the enzyme amylase is found in high carbohydrate fruits like apples and peaches. Fruits that are high in fat, such as avocados, contain the enzyme lipase. Further, we will focus on enzymes we obtain from different sources, that is animal, plant, mammalian cells and microbial cells, and also their respective usefulness and limitation. First, we will see commercial sources of enzymes. Biologically active enzymes may be extracted from any living organism. A very wide range of sources are used for commercial enzyme production. The different organisms and their relative contribution for the production of commercial enzymes are given below. Fungi 60%, bacteria 24%, yeast 4%, streptomyces 2%, higher animals 6% and higher plant 4%. A real breakthrough for large-scale industrial production of enzymes from microorganisms occurred after 1950s. Let's see enzymes from animal and plant sources. In the early days, animal and plant sources largely contributed to enzymes. Even now, for certain enzymes, they are the major sources. Animal organs and tissues are very good sources for enzymes, such as lipases, esterases, and proteases. The enzyme lysozyme is mostly obtained from hen egg. Some plants are excellent sources for certain enzymes, papain and bromelain. There are several drawbacks associated with the manufacture of enzymes from animal and plant sources. The quantities are limited and there is a wide variation in their distribution. The most important limitations are the difficulties in isolating, purifying the enzymes, and the cost factor. As regard as regards extraction of industrial enzymes from bovine sources, there is a heavy risk of contamination with bovine spongiform encephalopathy. BSE is a prion disease caused by ingestion of abnormal proteins. For these reasons, microbial productions of enzymes is preferred. Now let's see enzymes from mammalian cell culture. There exists a possibility of producing commercial enzymes directly by mammalian cell cultures. But the main constraint will the cost factor, which will be extremely high. However, certain therapeutic enzymes such as tissue plasminogen activator are produced by cell cultures. Now let's talk about enzymes from microbial sources. Microorganisms are the most significant and convenient sources of commercial enzymes. They can be made to produce abundant quantities of enzymes under suitable growth conditions. Microorganisms can be cultivated by using inexpensive media, and production can take place in a short period. In addition, it is easy to manipulate microorganisms in genetic engineering techniques to increase the production of desired enzymes. Recovery, isolation, and purification processes are easy with microbial enzymes than that with animal or plant sources. In fact, most enzymes of industrial applications have been successfully produced by microorganisms. Various fungi, bacteria, and yeasts are employed for this purpose. Now let's talk about Aspergillus niger, a unique organism for production of bulk enzymes. Among the microorganisms, Aspergillus niger, which is a kind of fungus, occupies a special position for the manufacture of a large number of enzymes in good quantities. There are well over 40 commercial enzymes that are conveniently produced by A. niger. These include alpha amylase, cellulase, protease, lipase, pectinase, phytase, catalase, and insulinase. So this is all about the commercial sources of enzymes, its usefulness and limitations. Thank you.